Good evening, everyone. So this week we'll be discussing embryology of the skin. In the previous multiple weeks, we have discussed the formation of keratin. We have discussed the formation of melanin. We've also discussed how is the process of melanin formation in melanocytes and then transfer to keratinocytes, and how are the keratin filaments are formed and they are aggregated in the developing keratinocytes as the cell migrates from the basal layer to the corneal layer. Okay. So I thought that if we have tackled the two basic processes of skin formation, that is pigment formation and keratin formation, we should also know about what is the developmental journey of forming a skin. What is the embryology of the skin? How do we get our epidermis? How do we get our dermis? How do we get our different appendages? And over the course of embryonal development, when the baby is developing from a single cell to a fetus and then an embryo and then eventually newborn baby, what are the processes that happen so that we get our epidermis, dermis, and other appendages like hair, nail, and eccrine glands, apocrine glands, etc. So this week, I thought that it is a very important, not a concept, but very important lecture, so that we'll be covering all the development of skin and skin structures through this video lecture. Without much further ado, we'll start our discussion on embryology of the skin. Let's move forward. Now, during the course of this lecture, we'll discuss cells and cell structures, which are the, keratinocytes or melanocytes and fibroblasts and all sorts of cells with the granular layer. So we'll discuss those cells, discuss their formation, and also the different structures like hair, nail, glands, and etc. We'll keep on discussing them. In signaling pathway, we'll be discussing about genes. I will just touch upon it a bit. I will not go into detail because those are not important. But we should know the major genes, two or three genes that are responsible for development of skin and its structures. After that, we'll start embryogenesis of skin. That means how the epidermis actually forms. So we'll do a bit of general embryology. For example, we will cover the stages from a two-celled fertilized stage to the formation of the three germ layers. And then afterwards, we'll continue with the development of skin. So after the three layers are formed, and the three layers majorly is the endoderm, ectoderm, and in between them, rather here, in between them is the mesoderm. So this is the three-layer formation, and we'll discuss how it forms. After that, we'll discuss how does the epidermis forms, how does the dermis forms, and what is the embryogenesis of other adnexal structures like hair and glands. Okay, so let's move forward. Now, just recapitulating the cells and structures of the skin, we have the keratinocytes, which are the main cells of the epidermis, and they're responsible for structural resiliency. That means to provide strength to the skin. The fibroblasts are main cells of the dermis, and they secrete collagen. The appendages like hair follicles, sweat glands, nail apparatus, these are the appendages. The melanocytes secrete melanin, absorb and block UV radiation, and provide skin color. The Langerhans cells are antigen-presenting cells, and they're present in epidermis to start the innate immune response. So whenever there is a pathogen and those pathogens come into contact with Langerhans cells, they will initiate a cascade of a process which will eventually give rise to or eventually will start the innate immune system, innate immune response. Other structures like sensory neurons, arterioles, venules, capillaries, lymphatics. So these are the other structures apart from the structures like keratinocytes or fibroblasts. Let's move forward. Now, in signaling pathway, the most important gene is WNT ligand or WNT gene. These are extracellular secreted proteins, the product of those genes, and they activate the frizzled receptors. Now, two major receptors that you will hear about is one is the smoothened receptor and other is the frizzled receptor. Okay, so WNT acts on the frizzled receptor. Okay, the function of WNT is to stabilize beta-catenin, and beta-catenin is a very important molecule that controls cell migration and proliferation. So that is the function of WNT beta-catenin pathway. Now, beta-catenin controls epithelial differentiation. That means in our body, there are different kind of epithelia. You have different epithelia in the oral mucosa, different in genital mucosa, you have different in the anal mucosa, different skin. This is totally different. So this differentiation is controlled by beta catenin because it is controlling cell migration and proliferation. That means it is responsible that what kind of cell will reach eventually where. Okay. It is also needed for appendage function. P63 is known as the central regulator of epithelium, and this could be a good Viva question or a good quiz question. Okay. Central regulator of epithelium is P63, and if P63 is absent, epidermis fails to stratify. There is no layering of the epidermis. Epidermis does not form layer. There are no appendages. So what do we mean to say is that P63 is an essential molecule to control epidermal differentiation. Okay. The fourth gene is EDAR or ectrodysplacin A receptor and EDA ligand. They are a TNF alpha family and they are responsible for development of the appendages. Clear? Let's move forward. The fifth is Notch receptor, and Notch receptor is responsible for epidermal differentiation. That means what is the eventual differentiation or what is the lineage of different epidermal cell layer? How does one cell continue to divide for certain time and after that it starts to differentiate and form specialized tissue structures. Okay. Now, BMP is bone morphogenic protein. It is responsible for regulating hair follicle cycling and the growth of hair follicle. We'll discuss them when we'll be discussing embryogenesis of hair. Second is HOX or homeobox transcription factor. It is responsible for body patterning. So body patterning means that in a developing embryo, what you have is just a disc initially. And this is known as embryonal disc. Okay, embryonal disc. Now, what part will form head? What part will form the tail? What part will form the left or the right part of the embryo? That is decided by the Hox gene. So, a proper functioning and activated Hox factor is responsible for the embryo to know what is the orientation of the embryo. Otherwise, it's just a disc, anything can form anywhere. Okay. Eighth is SHH ligands or sonic hedgehog ligands. These are extracellular secreted proteins and they bind to the smoothen receptor. Remember, I've told you two receptors. One is smoothen, other is frizzled. Frizzled is for WNT, smoothen is for SHH, sonic hedgehog. They are needed for hair formation and function. So remember, like you have a hedgehog and it has all the spines coming out from the back, like hair. So these are tightened hair or collected hardened hair in a hedgehog. So then you can remember that sonic hedgehog protein is required for proper hair function and development. Clear? Let's move forward. Now, with these two things out of the way, we'll start a journey and understand how is the skin formed, how is the epidermis formed. Okay. Now remember, in a developing skin, you have an ectoderm, a mesoderm, and an endoderm. Endoderm, mesoderm, ectoderm. Clear from outside to inside. Ectoderm gives rise to the epidermis. Underlying mesoderm gives rise to the dermis of the body except two areas. The face and the frontal scalp comes from neural crest cells. Okay, so dermis of the face and the frontal scalp is derived from neural crest cells, while the dermis of the entire body is derived from the underlying mesenchyme or mesoderm. Okay, neural crest cells also give rise to melanocytes. So I will not go into much detail about the embryogenesis of melanocytes. We have covered that in detail in the lecture on melanogenesis. So I request you to go back and see that segment of embryogenesis of melanocytes. Okay, let's move forward. Okay. So now we'll study how the cell layers are going to proliferate, divide, and form the epidermis. Okay. We have the two cell stage at day one. Okay, so you have a gamete. Okay, you have the ovum, you have the spermatozoa, and post fertilization, you have a zygote. Okay, now this zygote will divide into two cells, and this is the two cell stage at day one. Clear? On day two, you have the four cell stage. So what has happened? These cells have divided further, and now you have the four cell stage. At 3 days, you have morula. So morula means mulberry. Mulberry. It's a 12 to 32 cell stage. So what has happened is these cells have divided a lot and they have become a bunch of cells, roughly about 12 to 32 in number. And that stage at the end of 3 days is known as morula. Now at 4 days, all these cells which have divided in the center or nearly all the cells, they will migrate to a part or one end of the cell or one end of the collection. Some of the cells will form a lining. So this is the lining. These are all the other cells are collected at one end. So these are the collection. Clear? Now, when all the cells will go to one end, you will have a large cavity in between. Okay, it is not an empty cavity, there's fluid inside, but there are no cells. This is known as the blastocyst cavity. Clear? Now, in those cells, some of the cells which are now labeled in pink or skin colored are known as trophoblast. They will be responsible to create the placenta. Syncytial trophoblast and cytotrophoblast, they will be responsible to create the placenta for adequate exchange of nutrients from the mother. Okay. The cells which are labeled in blue are known as embryoblast, and these cells are responsible for the formation of embryo. Okay, so trophoblast will lead to placenta, will lead to attachment. Embryoblast will lead to embryo, lead to baby formation. Clear? At this stage, at the end of 4 days, it is now known as blastocyst. Let's move forward. Now, you have the blastocyst. Some cells of the embryoblast will mature to form anterior visceral endoderm. And this anterior visceral endoderm will decide what will be the pattern of axis or how the axis will develop, how the body axis is going to develop from here to here. Okay. Some cells labeled in yellow will form the layer known as hypoblast. Hypo means under. And some cells labeled in blue will form a layer epiblast. Epi means over. So above layer is epiblast. Below you have hypoblast. Now, when the cells are developing from day 4 to day 6, cells that will form hypoblast and cells that will form epiblast are mixed together in the embryoblast. But gradually over the next 1 or 2 days, they will migrate and reach downwards to form hypoblast and remain upwards to form epiblast. Clear? So both cells are mixed together initially and then they segregate. At about 7 to 8 days, the entire blastocyst gets implanted in the uterine wall. Okay, we know that the implantation occurs 1 week after fertilization. Okay, now when that happens, the lining cells and the trophoblast cells will multiply a lot because they have to form the placenta. Okay, for proper exchange of nutrients. So that has happened at the end of 7 to 8 days. Let's move forward. Now we'll start the process of granulation. So granulation means formation of the three layers, endoderm, ectoderm, mesoderm. Clear? So at 7 to 8 days, the implantation has occurred. The cell layer labeled in blue is epiblast because it is above or closer to the uterine wall, while the cell layer labeled in yellow is hypoblast because it is under or away from the uterine wall. Clear? Now, at 12 days, what actually happens is that these hypoblast cells, they will proliferate and line the entire cavity. Okay. Yes, between this lining and the outside trophoblast lining, this area will form multiple empty spaces. Okay. So the cavity lined by the hypoblast cells is known as the primitive yolk sac. Not the definitive yolk sac, the primitive yolk sac. Clear? Now these empty spaces which was between the hypoblast and the trophoblast layer, it grows larger. Okay, it connects together and grows larger into a very big cavity. Now this big cavity is known as the extraembryonic coelom. Clear? You need not remember all of this. I'm just telling you so that you can get some orientation what is happening. At 17 days. So now we are focusing on only this area. Okay, this area here. This I have enlarged. This area. So this I have enlarged. Clear? So what actually is happening? This cell layer, this layer which is surrounded by epiblast cells, it will also become larger. The cavity will become larger. So we have the cell lining now lining a cavity. That cavity or that lining will be now known as ectoderm. Same thing happens below, and that lining will be known as endoderm. And then you have the layer in between, which is intraembryonic mesoderm. The lower cavity is now yolk sac, while the upper cavity is the amniotic cavity. Clear? Now, one thing to remember and understand very clearly is that all the three cell layers, endoderm, ectoderm, mesoderm, arise from epiblast cells. Clear? So what actually is happening? Where is hypoblast cells? Okay. What actually happens is that epiblast cells will keep on dividing to form newer cells that will go downwards and replace the hypoblast cells. After they have replaced the hypoblast cells, the remaining cell will remain in between the hypoblast and epiblast to form the intraembryonic mesoderm. So, all the three layers, ectoderm, intraembryonic mesoderm, and the future endoderm, is made by only epiblast cells. Hypoblast does not give rise to these cells. Clear? Now, at the end of 17 days, the process of granulation is complete. Let's move forward. So let's understand it with a schematic diagram. At 15 days, you have an area which is the primitive yolk sac lined by hypoblast cells. You have the amniotic cavity lined by epiblast cells. Clear? At around 16 days, the cells from the epiblast will migrate downwards and multiply. These are known as invaginating epiblast cells, here labeled in green. So look at this picture from Langerhans. The blue is the epiblast, yellow is the hypoblast. Cells from the epiblast are migrating inside. Okay, if you can see here, the cells are going from here and going below, from medial to lateral. Clear? And you can see here that the cells are replacing the cells of hypoblast. So eventually what will happen is the entire layer will be replaced by the coming cells from the epiblast, and the remaining cells will remain between the epiblast and hypoblast to form the intraembryonic mesoderm. Okay, so let's move forward. Let's remove the markings and move forward. Clear? Now at 17 days, you have distinction of three layers. You have the ectoderm in blue, you have the mesoderm in red, and you have the endoderm in green. And diminishing yellow layer, which is the primitive hypoblast cells, but they will be replaced by the endodermal cells. And when that has happened, you have the yolk sac, not the primitive yolk sac, but the yolk sac. This remains the amniotic cavity. Clear? Let's move forward. Now, we'll see the development of neural crest cells. So this, we have reached this stage. We have the blue ectoderm, we have the red mesoderm, we have the yellow endoderm. Clear? Now what happens is in between a slight depression occurs, and on the adjacent side of the depression the ectoderm swells up. It starts to swell. So this is known as a neural plate formation. Okay, so it swells. This whole area, this whole swelling is known as a neural plate. Clear? Now at the tip of this swelling, hair and hair, like a wave, the topmost part of the wave is known as a crest. So the crest of the neural plate is known as neural crest. Cells populating this area are known as neural crest cells. Okay, so neural crest and these cells are the neural crest cells. So here you can see in the cut section, you can see the neural crest cells labeled in purple. Okay, eventually the central depression becomes deeper to form a groove, and now it is known as neural groove. The groove is deeper, and we can see that the neural crest cells are proliferating. They are multiplying and they are increasing in number and size. Clear? After that, the uppermost part will stick together and a tube will be left below it. This is the neural tube. The upper part has closed. Clear? So this was a groove before. The upper part has closed now to form a neural tube. Okay. The neural crest cells now lie between the ectoderm and the neural tube, in between them. Okay. Look at this diagram. So you have the ectoderm, you have the endoderm, and in between them you have the neural crest cells. Clear? So this is how the neural crest cells form, which will eventually give rise to dermis of the face and frontal scalp, along with melanocytes. Let's move forward. Now, we have formed the three layers: endoderm, mesoderm, ectoderm. So let's start the formation of epidermis. At about third week, the ectoderm forms neural plate and it invaginates to form the neural crest at the tip and neural groove below. Okay. Neural plate closes to form the neural tube. We know that. Neural crests migrate through mesoderm by utilizing WNT signaling. We know that WNT is responsible for migration of cells. These migrating cells will eventually form melanocytes and dermal fibroblasts of face and anterior scalp. We know that. If there's lack of migration, that means if the cells are not able to reach eventually in the epidermis, you have deep blue nevus, dermal melanocytosis, and Mongolian spots. Clear? So if any one of you know what are the examples of dermomelanocytosis, please mention in the comment section. Let me see. Now, this is the ectoderm. This is the neural tube. Clear? And these are the neural crest cells. Neural crest cells. These cells will migrate in a dorsolateral pathway and also in venteromedial pathway. Maximum migration of cells, which will eventually form melanocytes, they go through the dorsolateral pathway. Some of them do go through the ventromedial pathway. But major development of cells in venteromedial pathway will be glial cells. They will go to CNS. Okay, because this is the neural tube. They are going adjacent to neural tube. While the dorsolateral pathway majorly forms melanocytes. So glial cells and melanocytes have same lineage, same progenitor cells, but depending on what pathway they have taken, they will form different cell lines. Either they will form glial cells or melanocytes. Now, some melanocytes can also traverse through the venteromedial surface, may eventually reach CNS to form areas like substantia nigra and all that. Because we know that there are melanin deposits in the meninges also. Sometimes these meningeal melanin deposits can be a precursor to meningeal melanoma. Meningeal melanoma. Clear? So these kind of things can also happen because in embryology, the cells migrating from the venteromedial pathway will eventually form glial cell, but some migration of melanocyte precursors also happen. Okay, I request you to go to our lecture on melanogenesis, and I've discussed that in great detail. What is the embryology of melanocytes? Let's move forward. Now, neural crest cells derive the dermis in the face and neck and melanocytes, along with smooth muscle cells to blood vessels of the face. Clear? Ectoderm gives you epidermis, including the hair and nails, and subcutaneous glands. So I've just mentioned in bold what is needed for skin development, while all the derivatives I've mentioned here just for academic interest. Let's move forward. Hmm. Somites. So what actually happens is you have the, let me just change the color. You have the ectoderm, you have the endoderm, you have the neural tube, then you have what is known as a somite. So this condensation of mesoderm adjacent to the neural tube is the somite. Then you have the intermediate mesoderm. Intermediate mesoderm. And then you have lateral part which is divided into two layers, somatopure and splanchnoplure. No need to remember this, it's just embryology, don't worry. This is the lateral plate mesoderm. Clear? Now this somite, which is lateral to the neural tube, will form the axial skeleton and musculature along with dermal fibroblast of the body. These somites are also known as dermatomyotome. Dermatomyotome, why? Because they give rise to skin, dermis, and muscles. Okay, so that is why it is known as dermatomyotome. So this somite is the development of dermis of the body. Okay, dermal fibroblast of the body. Okay, let's move forward. Now this diagram I've taken from Fitzpatrick and we'll quickly go through it. We have the mesenchyme, which will eventually give rise to dermis, and we have the surface ectoderm, which is now only a single layer thick. Now this layer will divide into two parts, a superior periderm, which is a very thin layer, and a below germinative layer. Okay. Now other cells will keep on coming from the division of basal cell layer in between the periderm and the basal layer, and this third layer will be known as intermediate layer, which expresses keratin 1 and 10, which is similar to granulosum and spinosum in the adults. And at about week 12 roughly, you will start to see the four layers of the epidermis. Okay, so by the start of second trimester, you start to see the four layers of the epidermis. You have the development of epidermis, while the below the corium part, don't remember the corium part, just remember that this will form the dermis. You have the four layers available: basal layer, spinous layer, granular layer, and corneal layer. Clear? Let's move forward. Now, at 8 weeks, the epidermis begins to stratify with P63 expression. Keratin 8 and keratin 18 are the initial keratins, which eventually stops, and keratin 5 and 14 rises up, and these are the mature keratins in the basal layer. At 15 days EGA, periderm forms. Okay, periderm then starts to express 17 and 6, and the genes required for formation is SFN, IRFC, IKK through NFK beta pathway. You need not remember the genes, it is just there for academic interest. Now, periderm is responsible for transport, protection, and epidermal disadhesion. What do you mean by that? So when the limb buds, for example, if the arms has to form, so they will eventually form, let's say this is a developing embryo, they will form a bud, and then this bud will eventually keep on elongating, keep on elongating, keep on elongating, and eventually it will form an arm. Okay? And similarly, fingers. Clear? Now, what would happen is there has to be some protection to the developing bud so that it does not attach itself back to the epidermis. To prevent that attachment, periderm helps. So this is known as epidermal disadhesion. Now, periderm is shed into the amniotic cavity and also forms a major part of vernix caseosa. Vernix caseosa is a cheesy material lining the newborn. Now, this periderm undergoes terminal differentiation to form mature keratinocytes. Sorry, the developing epidermis undergoes terminal differentiation to form mature keratinocytes. And at the start of second trimester, cells in the intermediate layer have formed granular and spinous layer, or at least commit to form. That they will eventually form the granular or the spinous layer. Let's move forward. At about 22 to 24 weeks, the epidermal cornification begins, first on the head, palms, and soles. So these are all pressure-sensitive areas, thicker areas, scalp, palms, and soles. So cornification starts first on these areas. At 24 to 26 weeks, stratum corneum is many cell layer thick. At mid-third trimester, the epidermis is same as adult skin, but proper barrier function will continue to develop few weeks after birth. If there is a disorder earlier, before the layers have started forming, it is incompatible with life. But if it occurs later, it can give rise to mosaic disorders. Now, remember what I'm trying, just understand what I'm trying to say. If a cell is migrating to a certain part of the body, and before migration, the cell is mutated and it is not able to carry out its normal function, the cell will die, and that will be incompatible with life. But let's say if the cell is migrating and during the migration the mutation has occurred, so all the cells supplied in that tract will be mutated. So at birth, you will have problem in a pattern, and that is known as mosaic disorders. Clear? So if the disorder occurs early, then it is incompatible with life. If it occurs late, then it leads to mosaic disorders. Let's move forward. So this is the same thing from Bologna, which we are trying to understand, that this gray layer is the ectodermal layer. Okay, this gray layer is the ectodermal layer, and eventually it will divide into a thinner periderm and a basal layer, which is now known as germinative layer. And then all the four layers, which is the intermediate layer first, which will eventually give rise to granular and spinosum layer. You have the basal layer and the corneal layer. Okay, so at 36 weeks, you have the superficial periderm layer developing from the basal layer. At 72 days, you have the intermediate layer, and at the start of second trimester, you will have distinct four-layer structure of the epidermis. Let's move forward. Now, embryogenesis of melanocytes. I will not go into much detail to this because I've already covered it. That in the that part has already been covered in melanogenesis. So just go back and look for the videos there on the channel, and you will understand the melanogenesis of melanocytes. Sorry, embryogenesis of melanocytes. So at about 50 days of EGA, HMB45 immunostaining is seen. So it's a special stain to look for melanocytes. Bologna mentions 8 week and 50 days is mentioned in Fitzpatrick. So I've mentioned both of the areas here. At the starting, the melanocyte density in the epidermis is about 1000 cells per mm square. As the cell layer begins to divide, the melanocytes will also divide. So at 80 to 90 days EGA, it is about 3000 cells per mm square. Late, all these cells will start to atrophy, start to destroy themselves, and only 800 to 1500 cells remain per mm square, which is roughly equal to what we see in adults. Okay. Now, you have a progenitor cell under the action of SOX10. So SOX10 positive cells will form another precursor cell. Okay. So this will be a precursor cell. Now, this precursor cell, if it gets activated by MITF, microphthalmia associated transcription factor, it will become a melanoblast. And if this activation doesn't happen, otherwise you have, let's say, a SOX9 or some other kind of mutation, sorry, activation. If that activation, MITF activation doesn't occur, then it will lead to glial cell. Okay, so MITF is a very important gene that regulates melanocytes, and we have covered them in great detail in the video on melanogenesis. So go and look at that, and you'll be able to understand what exactly is happening here. Let's move forward. Now, melanin production starts at about 3 to 4 months, and the transfer starts from 5 months onwards. But still, after birth, melanization increases. So there have been instances when the child is born quite fair, and eventually, over the neck of the succeeding few weeks, the skin slowly becomes darker. So parents usually come and complain that my child was born fair. I don't know what has happened. The child has become darker because melanization process will keep on continuing. Okay, for a few weeks after birth. Now, the dermal melanocytes. Remember, the melanocytes are migrating from a dorsolateral pathway and a ventromedial pathway, and they will eventually reach the dermis, and from dermis to the epidermis, they will go. If some cells remain in the dermis at birth, most of these cells will either go to the epidermis or they will atrophy. If they do not atrophy, or if they are not able to go towards the epidermis at birth, this will lead to formation of dermal melanocytosis. Okay, again, if you know the examples, mention in the comment section. Let's see. Let's move forward. No, embryogenesis of dermis. We have completed the development of epidermis. Let's go to dermis. The neural crest ectoderm will give rise to the dermal mesenchyme of the face and anterior scalp. So neural crest somewhere here will give rise to the dermis of face and frontal scalp. Okay. The embryonic somite. So this area, let me change the color. This area which is just adjacent to the neural tube is the somite, which and the part of somite that forms dermis and muscles is dermatomyotome, and this will give rise to the dermal mesenchyme of the back because that is the eventual area of this formation, somites. So it is the axial back. So this area, the dermis will be formed by the somites. Clear? Now lateral plate mesoderm. So the one labeled in green here or labeled in black now is the lateral plate mesoderm, which will give rise to the dermal mesenchyme of extremities and ventral trunk. So this is the ventral trunk. Okay. So that is how dermis is derived from different sources. Okay. And these mesenchymal precursors will eventually give rise. Mesenchymal precursor means the cells which will form all other cells in the dermis. They will give rise to papillary dermal fibroblast, reticular dermal fibroblast, and adipocytes. And for that, you require the Hox gene, which is responsible for patterning. So, some precursors will form fibroblast, some precursors will form reticular dermal fibroblast, and from reticular dermal fibroblast, you have the eventual formation of adipocytes or fat cells, and they will eventually go to the subcutaneous. Clear? Let's move forward. Now, embryogenesis of fibroblast. At 6 to 8 weeks of EGA, you have presumptive dermal fibroblasts under the epidermis, which can form any tissue. So you have the presumptive fibroblast below the epidermis. Simple fibers are secreted. Collagen 3 is to collagen 1 ratio is 3 is to 1. Okay, so collagen 3 is to collagen 1 is 3 is to 1, which is reverse of adult skin. We know that in adult skin, collagen 1 is major collagen, while in the embryo, it is collagen 3. Gradually, this will reverse and form the same as adult. Now, we know that the fibroblasts are going to be different. Why different? Because the source of dermis is different. So in head, the fibroblasts are derived from neural crest cells. In dorsal fibroblasts, that means of the back, they are derived from somites and dermatomyotomes. And in the ventrum, they are derived from the lateral plate mesoderm. Why? Because the dermis is derived from different areas. Let's move forward. Okay. At 12 to 15 weeks of EGA, this fine papillary dermis is different from the thicker, deeper reticular dermis. So this kind of stratification occurs. Papillary dermis will be different than reticular dermis. Complex fibers emerge. The upper dermal fibroblast, which are BLIMP1 positive, DLK negative. You need not remember it. I'm just mentioning it for academic interest. Upper dermal fibroblasts, which are BLIMP1 positive and DLK1 negative, will give rise to papillary dermal hair and erector pili. Why? Because these are the structures in the above half of the dermis. Lower dermal fibroblasts, which are BLIMP negative and DLK1 positive, will give rise to reticular fibroblasts and adipocytes. So upper dermal will give rise to papillary dermal fibroblast, hair and erector pili. Okay, hair and erector pili. While in lower dermal fibroblast will give rise to reticular fibroblast and adipocytes. Clear? Gradually, the gelatinous proteoglycan-rich cellular dermis will be replaced by a rigid fibrous paucicellular dermis similar to that of adult. So proteoglycan-rich gelatinous dermis will replaced by a hard fibrous paucicellular adult dermis. At the end of second trimester, the dermis will change from non-scarring to scarring dermis. Why? That is, after second trimester, if any trauma occurs to the dermis, it will heal with scar formation. It will heal with fibrosis. If any trauma occurs before that, there will be no scarification or no fibrosis. That is what we mean to say. At about 22 to 24 weeks, elastic fibers emerge. So elastic fibers starts to get formed. Let's move forward. Now, for vessels, nerves, and fat, this is the easiest part. At the end of first trimester, a basic pattern has been formed. So we have the pattern A basic pattern of arterial differentiation branching has been formed. Along these branches, angioblasts, which are the precursor cells, will accumulate, they will collect, and they will proliferate to form rods. Eventually, these rods will start to form a lumen in between, okay, which will grow bigger, and you will have a tube, and that is how your capillaries and arteries will form. Extensive remodeling occurs in utero till after birth. The control is by these ligands. These are just for academic interest. Developing nerves dictate arterial differentiation and branching. So, multiple times it has been mentioned that the growing nerve will dictate how the adjacent artery is going to branch. Now, you have venous endothelial cells. Venous endothelial cells, if they are active upon by the PROX1 gene, this will lead to development of lymphatics. If the PROX1 gene is not acting, then it will lead to formation of venules. Clear? Just remember that PROX1 gene is responsible for lymphatic formation. Let's move forward. Now, adipocytes or the fat cells are derived from the lower dermal fibroblast progenitors. We know that. At the level of second trimester, subcutaneous fat accumulation starts. So what I'm trying to say is this is the epidermis, this is dermo-epidermal junction, this is the dermis. From the lower part, slowly adipocytes will be formed. And these adipocytes will proliferate and go deeper. Clear? By the end of third trimester, the dermis will invaginate inside in septa. And these will form septa. Okay? And eventually you will see the adult pattern of septolobular subcutis. Okay, so this is a lobule while this is a septa. So septolobular subcutis is formed like that. Just remember that adipocytes are derived from lower dermal fibroblast progenitors. Clear? Let's move forward. Now, embryogenesis of dermo-epidermal junction. It is very easy. From in the embryo, dermo-epidermal junction, lamina lucida and lamina densa is already present. So two layers are already there. Gradually over 8 weeks, specific components start to appear. So like all the components of hemidesmosomes, desmosomes, placophilin, placoglobin, everything which we have already discussed in the basement membrane zone series, all the molecules will start to develop. Anchoring fibrous, anchoring filaments, everything will be there. At about 12 weeks, you have the mature components of dermo-epidermal junction. So at 8 weeks till 12 weeks, this layering starts and the four-layer structure of the dermo-epidermal junction is there. And with that, with progression, with ongoing time, with passing of time, you will have this four-layer structure will be thrown into folds. Why? Because remember the dermo-epidermal junction is in folds, papillary dermis and retipex. So this fold will gradually develop in all the four layers. That is how the dermo-epidermal junction is formed. Just remember 2 points, 8 weeks and then 12 weeks. Clear? Let's move forward. Now, we have formed epidermis, we have formed dermis, and quickly we are going to form the hair follicle now. So dermis stimulates epidermis to become focally thicker and will become placoid. And this requires WNT signaling. We have discussed that before. This happens at about 75 days at gestational age. Now these placoids are first formed on scalp and face at about 11 weeks. And then they go downwards to caudal and ventral direction. Clear? So like this caudally. Clear? Now this placoid will stimulate the dermis to form dermal papilla. So first, a group of dermal mesenchymal cell will stimulate the above epidermis to form placoid. This placoid will stimulate the lower dermal mesenchymal cells to form dermal papilla. This dermal papilla will again stimulate the placoid to go inside to proliferate and become hair germ. Now this hair germ will keep on elongating to form hair peg. And as soon as that hair peg is formed, inside the hair peg, the shaft will start to keratinize, and as soon as that starts, it becomes a bulbous hair peg. Okay. So at about 80 days EGA, hair germ is formed. At about 100 days EGA, hair peg is formed. At the bulbous peg stage, sebaceous gland formation starts, according to some authors. So what actually happens is you have the placard in the epidermis, then you have the hair germ, then you have the hair peg, and then you have the bulbous hair stage when the hair shaft has started to grow. Clear? Now there is a superficial bulge above and there is a deep bulge or a deeper bulge. This superficial bulge will give rise to sebaceous gland. The deeper bulge will give rise to attachment of erector pili muscle. It also has follicular stem cells. Clear? At about 130 days EGA, lanugo hair develops. So these are the first hair that develops in the embryo, and they are shed before birth. In fact, shedding happens 1 or 2 times before birth, and from the third cycle onwards, the hair become asynchronous. What do you mean by that? That the hair cycle is now at different times. They are not in an orderly function, they are disordered. That is why you don't lose hair, you know, you do not lose all your hair and then grow again and then lose your hair, because the cycles are in different stages of time every time. Okay. I've mentioned some genetic controller here, but that is just for academic interest. If you want, you can just have a read. Just remember that BMP inhibition is required and WNT enhancement is required for proper hair follicle development. Clear? Let's move forward. So this diagram I've taken from Fitzpatrick to explain the development of hair follicle. We know that the dermal mesenchymal cells stimulate the epidermis to form a thicker area which is known as placard. Now this placard will keep on growing and will stimulate the dermal mesenchymal condensation to form the dermal papilla. So here the dermal papilla is slowly forming while the placard cells are dividing and going inside. So a hair germ is a spherical structure while a hair peg is more of a cylinder. Okay. Now when the hair shaft starts to develop and the dermal papilla has invaginated, you have the development of a superficial bulge and a deep bulge. A superficial bulge will give rise to sebaceous gland, and deep bulge will give rise to the attachment of erector pili. Clear? Just remember this. Let's move forward. Now, at second trimester, 7 concentric cell layers from outside to inside are formed. Ovaries, iris, Henle, Huxley, cuticle, hair shaft, cuticle, cortex, medulla. Okay. At 18 to 21 weeks each year, the hair canal is fully formed and the hair starts to grow. The hair cycle starts. And this hair cycle will become asynchronous, that means out of order, after birth. Proper hair cycle starts from the third cycle perinatally, that means around birth. Now, embryogenesis of sebaceous gland. That's easy. At 13 to 16 weeks EGA, most superficial bulge. Under the action of SOX9, LRIG1 hair keratinocyte stem cells, in 13 to 16 weeks, the superficial bulge, the most superficial bulge, will become a presumptive sebaceous gland. The outer layer of this gland will become lipogenic cells and start to form lipids. Okay, this lipid will keep on increasing in quantity after the adrenal gland. Remember, adrenal gland is formed in the body because other organs are also being formed. After the adrenal gland is formed in the body, under the action of adrenal hormones, further development is continued. So the first development is started by SOX9, LRG1, and dermal hair keratinocyte stem cells, but after the adrenal gland is formed, the adrenal gland takes over the development of sebaceous gland in the second and third trimester. Clear? Gradually, what will happen is the sebaceous gland duct will get canalized, will form a lumen, and will start to secrete the lipid secretion inside the follicle. Inside the infundibulum. Clear? And this contributes to vernix caseosa, the cheesy-like covering. The other contributor was periderm. Okay. Genes require a low activity of beta-catenin, increase CMI, increase androgen receptors, and increase P53. That is just for academic interest. Clear? Let's move forward. Now, embryogenesis of eccrine sweat glands. At 55 to 65 days EGA, large mesenchymal bulges or pads develop on palms and soles. Okay. At 12 to 14 weeks, which is roughly, this was, you could say approximately 8 to 10 weeks. 8 to 10 weeks. At 12 to 14 weeks, parallel ectodermal ridges develop overlying these pads, and these ectodermal ridges gives rise to the fingerprints. Fingerprints. Now, these mesenchymal pads will regress by third trimester. So, what actually happens is, in the epidermis, this becomes thicker. Then you have the formation of the ridges, and then at the end of third trimester, this thickening is resolved. And this thickening is why, because if you remember, for example, in dogs and cats, you have paws, you have those thicker areas. So that is what the epidermis is trying to form. But humans don't have that thicker area, so it regresses back and forms flat. Okay, so and this becomes fingerprints. Now, in between them, a part of the epidermis will invaginate inside gradually and then will start to coil. And this coiling will form the eccrine gland. Clear? Now, as the cells are migrating, the dermal component. So this is the epidermis and this is the dermis. The dermal component of the duct will canalize first, and the epidermal component will canalize afterwards. Clear? Now, at 14 to 16 weeks of age, individual eccrine glands and primordia bud, along with ectodermal ridges at regularly spaced intervals, they go inside and start to coil. So buds elongate as cords of cells and enter the pad mesenchyme. Just don't get confused. Remember, a part of epidermis will go invaginate inside into the dermis and starts to coil. At 16 weeks EGA, glandular structures are developed and the eccrine duct of the dermal component, that means the part of duct which is inside the dermis, starts to form a lumen. At 12 weeks, roughly 6 weeks after the dermal duct development, you have the development of epidermal duct. So, epidermis, dermo-epidermal junction, dermis, eccrine gland like this, coiling. So this dermal duct component, which is from here to here, dermal duct component will canalize first, and after 6 weeks, the epidermal duct component. Okay. The epidermal duct component will canalize first, farther. Okay. I'll show a diagram in the next slide. You will understand better. Clear. So at 5th month, the fingerprints are complete because the ridges are complete. The development starts in the 7th week approximately. At 7th month, the clear cells and mucin secreting dark cells of the apocrine glands are also formed. Now remember, apocrine glands are being formed from in the hair follicle. Clear? Now, so at 7th month, your clear cells and mucin secreting dark cells of the apocrine glands are formed, but that is just I've added here. Now remember that the sweat glands at about 12 to 13 weeks EGA will first involve the palms and soles. Because these are the sweaty regions, palms and soles. So first the development will happen over palms and soles, and at 20 weeks you have development over whole body. Clear? Let's move forward. So this is the diagram I was talking about. You have the pore. You can see the epidermis, and at the basal layer of the epidermis, a bud will form. This bud will further elongate downwards, and as it is started to coil, the lumen starts to develop at 16 weeks. And at about 22 weeks, the epidermal component also starts to develop, while the cells are undergoing a lot of coiling in below while developing into secretory cells, and these secretory cells will start to form sweat. Clear? Let's move forward. So with that, these are the reading recommendation. But if you've gone through this video in detail and making your own notes, you will have a good understanding of embryogenesis of skin and skin structures. Okay, let's move forward. A shameless plug time. Do consider channel membership. We have more than 120 members now, and they have access to better quality pictures, color clinical pictures, because I cannot use color clinical pictures on YouTube. Trust me, I have to upload a video 3, 4 times till the YouTube approves it. So it's creating a lot of issues. So members have access to colored photographs. They have access to a detailed, longer videos with extra information, with extra explanations. So if people are already members of channel, then they know that they are having access to other information that the public have. They don't have that. The video is shorter for the public. So, and I am planning to do some kind of case presentations. I just don't know how to get about it, but I am planning to do it. And Viva questions, exclusive videos, dermoscopy videos. I'm planning a lot of things which will first be released on the membership platform, and then depending on the response it gets, it will be released to the public. So more topics will be coming up every week. I'm trying to change and make some sense from one topic to another topic. What all topics to be covered, a general area to be covered. I'll be asking you people again. And the membership is just at about 59 rupees a month till this month. Okay, till the year ends, and from next year it will be increased. So consider becoming a member. It takes a lot of effort to create these kind of lectures every week, and this just gives me a further motivation to work and to make an hour-long lectures week after week after week. So if 60 bucks is nothing, I think you won't even get a good quality packet of chips for 2, 3 days only for this bucks. So if you can consider, do consider becoming a channel member. So with that being said, I shall be waiting for you to become a member. Let's move forward. So we'll finish the lecture now on embryogenesis of skin. We have covered a very wide area. We have covered general embryology. We started from two-cell stage, and we have reached by developing hair, nail, all the 4 layers of epidermis, all the dermal layers like papillary and reticular dermis, and subcutaneous vessel nerves. We have created everything. So now we have done the most basic part of basic dermatology, that means construction of skin. Clear? So I hope that you have understood. Usually, occasionally, there are short notes and exams on the regarding the embryonal development of skin. So we should have a certain idea. And if you just spend an hour on this video, you will understand what exactly is happening from where. If you have any doubts, any comments, any suggestion, mention them in the comment section. Till then, we'll stop this lecture now. Adios and bye bye.